morning, ladies and gentlemen. So I'm Anshala Kurupu and I'll be moderating this session today. And first of all, I warmly welcome all of you to this webinar on dengue. As you know, dengue is a huge burden in the world and especially in Asia. And today our topic will be on human immunity to dengue virus infections. At the end of the talk, we will have a brief Q&A session for about 10 minutes where you can ask any questions you have. And you can also use the chat feature in the Zoom if you have any questions. Now, this webinar is jointly organized by the Young Scientists Forum, or the YSF, and the Institute for Combinatorial Advanced Research and Education, which is also known as KDU-CARE, of the General Sir John Kothalavala Defense University. The Young Scientists Forum has been initiated by the National Science and Education Commission of the Ministry of Education, and this forum has been established to give younger people an opportunity to voice their opinions on science and technological matters, and also to develop their skills in sciences. The YSF actually holds frequent awareness programs and webinars to disseminate knowledge on important scientific topics. And if I give a brief introduction to KDU-CARE, or the Institute for Combinatorial Advanced Research and Education, so we are the research arm of the General Sir John Kothalavala University, and KDU-CARE facilitates advanced scientific education and research, where it aims to provide solutions to national and international industrial challenges. We also have many opportunities for postgraduate students like MPhils and PhDs at KDU-CARE. So now let me introduce our speaker for today. She is Dr. Anmol Chandle. Dr. Chandle has done her PhD from the National Center for Cell Sciences, Pune, India, which has been followed by a postdoctoral training at the Yale University New Haven, CT, USA. She is currently the group leader of the ICGB Emory Vaccine Center. During her research career, she has been deeply interested in signals and factors that dictate the formation of immunological memory and why, in certain cases, the body fails to generate long-lived immunity. The focus of her current research is about understanding human immunity to infectious diseases that are of public health importance to India, with a special emphasis on dengue virus infections. Today, she will share work going on in her lab toward the cellular B-cell responses and humoral immunity to dengue virus in children from India. And now I cordially invite Dr. Chandle to begin the talk. Over to you, ma'am. Thank you.
Thank you, Angela. It's a pleasure to be speaking to all of you. And thank you so much for inviting me to give this talk. And Angela, I hope you're having a good time in Delhi. And I hope there are some students in the audience, and please do avail the sort of fellowships ICGB provides. It'll be nice to have you come to Delhi and come to ICGB and do some amount of research. So do feel free to either speak to Anshala or email me if you want any more information about the kind of training that ICGB gives. We've got some wonderful scientists here, and I hope some of you can be a part of our ICGB community. So as Anshala has so nicely introduced me, I'm an immunologist, and most of my work is on infectious diseases. Viral infections are my particular favorite. I did not know my audience today. I kind of got a hang that maybe it'll be students, postdocs, but I'm not sure. So I've tried to keep the talk a little simple, but at the same time, give you a good flavor of immunology, especially B-cell immunology. Please feel free to stop me and ask me questions if something is too much. I've tried to put as many cartoons of the silliest things like how we do ELISAs. So it kind of, everyone's on the same page. So let me just start so that we don't take more than the requisite amount of time. So as Angela already said, dengue virus infections is a large program in the lab. We are really interested in it for multiple reasons. And this slide will actually give you an idea as to why. I don't have to introduce dengue to at least this audience because Asia has dengue and that's the bottom line. But India has a huge burden of dengue. The global burden is obviously there, but India, at least by certain figures, has about 30 to 50 million exposures or infections, depending on how you calculate it. And these infections are obviously resulting in certain level of morbidity and mortality. So there is a question right there to address. The disease itself, as I said, is a viral infection. This viral infection can be caused by 4 different serotypes, as we call them, of the viruses, which are aptly named as dengue 1, 2, 3, 4. The thing that you must remember about these serotypes for the rest of the talk who are not familiar with dengue as a virus is that these serotypes are about 60 to 65% ballpark similar. It's the 25, 30, 35% dissimilarity which actually is beneficial or not beneficial depending on which side of the coin are you standing, whether you're standing on the host side or the virus's side. So you can get infected by one serotype and develop long-lived immunity to that serotype, at least based on current standards. Heterologous infections, you are immune to them for about a couple of months to about a year, after which you get susceptible to them. I will come to the other point about homologous heterologous infections in just one second. Once you get infected, you may not even develop disease depending on how lucky you are. But when you do develop disease, now this disease can be across a spectrum. It could be mild disease, a febrile differentiated fever. You could be home for 2 days. You could have moderately severe symptoms where you may need to take a paracetamol or 2. Or you could develop severe symptoms, which could be hemorrhage or shock. And some of these infections can also be fatal, especially in children. But if you look at the graph, it looks something like this with DHF, DFSS, as we call it, dengue hemorrhagic fever, shock syndrome, falling at the tip of this pyramid, but that pyramid is enough, especially since it affects children. Now, this entire thing is very complicated, both from the virus side and also from the immune response. And there's not a lot understood in terms of why someone remains mild, why someone becomes severe. To compound all of it, we really don't have any universal vaccines. One of the forerunners was licensed in a few countries, but that's about it. We also don't have any antivirals or any prophylaxis in terms of monoclonals that are approved. Now, all this ties into trying to understand what causes severity. And one of the things, at least immunologically, that is of concern is that there is a concept of primary dengue versus secondary dengue. So, for example, if I get dengue 1 for the first time, it's my primary exposure to it. And if I get 2, 3, or 4 a later time, that's called secondary dengue. So that primary versus secondary dengue seems to play a large role with the idea that secondary dengue has a propensity to push you towards this side of the disease spectrum. So what do we mean by primary or secondary dengue? In fact, what do we mean immunologically? A simple slide for it. So when there are naive B cells in our body, when they see a virus or an antigen, they get activated, they form plasmablasts. Now, these plasmablasts are basically effector B cells. They are blasting, they are proliferating, and for the most part, they are antibody secreting cells, and they make tons of antibodies, apparently 1 million antibodies per second. So if you look at it in terms of timeline, if you have a primary infection, the first antibody response that you will make is an IgM response, and that over time will die down completely. Please don't look at this scale as exact. This is a cartoon, so don't take it on face value. This could just die down 7 days, 10 days, maybe even a month, depending on the infection. At some point of time, there's class switching that happens, which most of you would have heard of. And the class switching typically in a viral infection would happen to an IgG response. And at some point of time, the IgG response takes over. So this is typically what would look like in a primary infection. If you do get re-exposed at some point of time, you have memory cells that are already sitting around. So those memory cells are the ones that would take over, because memory cells are supposed to do rapid recall. And these memory cells get converted to ASCs, those antibody secreting cells. And obviously, since they're already class switched, they make IgGs from the get-go. So in terms of primary secondary responses, this is the window during the acute febrile phase that we look at. And based on the WHO criteria, we say that if IgM is greater than IgG during the febrile phase, you are a primary infection. There are ratios, but we won't go into it. And if your IgG is greater than IgM, then you are a secondary infection. This is how you diagnose primary and secondary infections. So where do we come to the idea that secondary infections are more severe? One of the first studies, really historical studies that were done, was actually done by Sabin. It's a wonderful study. Albert Sabin is of the polio fame. All of us know him. There's no introduction required there. And what he did was that he infected individuals with, at that time we knew of only two serotypes, so we'll call them 1 and 2, with one of the two serotypes. And what he did, he infected them across a few months to about a year. And then he came back afterwards, and some of them, he gave them the same serotype back again, which was their homologous challenge. And the other ones, he gave them a heterologous serotype. And what he noticed was that the people who got the homologous challenge again, suppose it was dengue 1 and they got dengue 1 again, it did not depend, they did not develop any infection. Whether he challenged them after 3 months or 6 months or even up to a year, they remained protected. Whereas people who got heterologous challenge, perhaps dengue 2, since there were only two serotypes known, they were okay for a few months, after which, as they were out by about a year since their exposure, they actually not only developed an infection, they actually developed severe infection. So one of the first things that noticed that secondary infections may be more severe was this study. After many years, this was followed up with why this could be the case by Dr. Halstead. All of us would have heard of him. And he gave a process due to which secondary infections may be more severe. And this was a concept called antibody-dependent enhancement of infection, which essentially is that they are cells that have Fc receptors on their surface. Fc receptors, as you would imagine, actually bind to the Fc portion of the antibody. Now, if you have antibody which is in subneutralizing amounts, it can still bind to the virus, but it cannot neutralize the virus. For example, if this particular virus required 10 antibodies, but I have only one antibody, this antibody will bind to the virus, but instead of neutralizing it, it can dock on Fc receptors and actually allow enhanced uptake of virus into these cells. And then the virus multiplies, and then you have increased viral load. Increased viral load means increased cytokine profiles, and that perhaps results in severe disease. So this was all well and good till we started to do some amount. Of course, there are lots of gaps in our understanding of the immune response, barring what I have just shared with you. So we were just looking at all sorts of parts of the human immunity, and we made some very interesting and surprising observations. So what we did was that we had already looked at some 600 odd children. First thing we found is that about 30 odd people, if you would focus on the red part of this pie, were severe dengue. These are hospitalized children in Delhi. So about 30% of them were severe dengue cases, and the rest of them were either DI or DW, which is dengue without warning signs and dengue with warning signs. But when we divided this pie amongst primary and secondary, how many were primary, how many were secondary, we were kind of hoping that this red would completely segregate into secondary dengue. But that did not happen. When we divided this pie into primary and secondary, based on the IgM-IgG ratio, what we found that this pie, this red part, remains mathematically more or less the same. So the conclusion of this was that basically, at least in India, we seem to see a significant proportion of primary infections are also severe disease cases. So while we are not surprised that secondary infections are severe, we actually found it very surprising that we see almost equal proportion of primary infections are severe. So one of the overarching questions that came in the lab was why is there severe disease in primary infections? So the way we are looking at dengue in our lab is in looking across the board at a lot of different aspects of human immunity. So we have a lot of collaborations across the country, and we get blood samples, peripheral blood from them. And all the samples that we get are already lab confirmed in the hospital, either by NS1 or IgM at that point of time, or there is retrospective PCR done. So we have divided our studies into innate immunity, T cell responses, and B cell responses. In innate immunity, we are doing a lot of things like looking at cytokines, MDSCs, and monocytes. In T cells, we are looking at their activation, their phenotypes, looking at what epitopes they respond to, what's their functionality, and we've also done some level of transcriptomics. And B cell responses, which I'll focus on today, we've done a plethora of things there too, looking at plasmablasts, which are those antibody secreting cells. What is their functionality? What does their RNA look like? What is their repertoire like? And we also make monoclonal antibodies to be able to individually see what each antibody is doing. When we do all this work, we get a lot of other information like clinical information, virology, and obviously we do a lot of serum studies of IgM, IgG, the isotypes, glycosylation patterns, etc. And all this gets wrapped together in the hope that someday we'll be able to crack some amount of this puzzle, obviously not alone, but we will get a little bit better understanding of human immunity to dengue. So what I'll do is I'll focus a little bit on the antibody responses and B cell responses and kind of rush a little bit from this introduction onwards. I wanted to remain slow on the introduction in case people were not immunology people or dengue biology people, we would all be on the same page. So here's our approach. Part of the approach I've already described. Take dengue patients, they are either PCR, NS1 or IgM confirmed. We isolate PBMCs, we isolate plasma, we do our ELISAs here and we do neutralizing antibodies. And on this side, we look at cellular responses, either plasmablasts or the functional responses. So one of the first questions we asked was, what's the magnitude of plasmablast response in dengue? And does it differ in children with primary and secondary infection? So here is a slide to show you what the plasmablast response looks like. And I'll quickly walk you through it. These are healthy individuals, basically no dengue. These are dengue febrile children. And this is the same person once they recover. This is anytime after a month. The way we gate on plasmablasts is we look at total B cells. So CD20 and CD19 are two markers for B cells. So all these are B cells. And then taking this, if we look at the cells that are either CD27 and CD38 high, those are the plasmablasts. So when we put all the individuals we've looked at, that's what it looks like. So healthy obviously have little to no plasmablasts. This is because we are not sitting in germ-free environments. We always have some level of antibody secreting cells, which kind of more or less goes down to normal by the time you recover. And then the individuals who have dengue febrile infection can go anywhere from 0.2% of total B cells all the way to 80% of total B cells would be a response. So it's heterogeneous because we are humans, but it is quite massive, which is very impressive as compared to, for example, flu or even chikungunya viral infections. This expansion of plasmablasts that we see in dengue probably is as similar to what people have reported for example, for Ebola infections. Now, if we segregate these individuals into primary and secondary responses, here's what is interesting. We actually do see that primary infections have lower plasmablast responses as compared to secondary infections, as you can see, based on the statistical significance here, which is nice. The second question we asked was, what is the functional difference in the antibody responses in primary and secondary? Plasmablast responses just gives a phenotype of a cell, that these are antibody secreting cells that are 2738 positive, but are they actually making dengue specific antibodies is the next question. So we can do this in multiple ways, but one way that we do it is look at everything across the board. So we look at the isotypes, we look at binding and avidity, cross-reactivity, neutralization, and even antibody-dependent enhancement. While we can do this in sera, and we can also do it at the level of the cells by doing ELISpot assays, but what will happen is that the serum gives you what you already had previously and what is ongoing, and there is no way to tease the two things apart. So the best way to address these questions is actually to look at single cell responses and to actually ask what's happening if I take confirmed primary people and confirmed secondary people, take their B cells and ask questions at the level of a B cell, because then you can ask questions like isotype, for example. So here is our approach. We actually make human monoclonal antibodies in the lab to actually ask what individual B cells are doing. And a quick way that the way we do it, at least in this study, it has been done in the traditional way. We do a single cell sorting of these plasmablasts. So every single cell falls into a well. This well contains capture buffer. The capture buffer instantly lyses the single cell that has gone in and the RNA gets released. And at that point of time, we do a series of cloning and then we divide the cell into half, and half the cell we clone out and express the heavy chain, and the other half of the cell we express the light chain, and then we bring it back in a transfection system. And then when we co-transfect these two plasmids that contains the heavy chain and light chain from the same cell, you get back the antibody that individual cell was making. And then we purify it just the way protein purification is done. We run it on a gel, and we should make sure we have an antibody that has a heavy chain and light chain, and it's not only the heavy chain or only the light chain that is getting expressed. And that way, when we do a lot of cell sorts and then we make a lot of monoclonals, we are actually able to look at hundreds of B cells at a level of their single cell functionality. So this slide real quickly, every time some panels are colored in this reddish orange color, they will always be secondary dengue. Whenever you see this bluish color, those are primary dengue. And this is how we do it real quick. Obviously, we want to take live cells, lymphocyte gate. This is our total B cell gate. Those are our plasmablasts. These have gone into 96 well plates. We do a purity sort to make sure that we are looking at only plasmablasts, and the below panel basically shows if you rerun those cells back through the sorter, you have a pretty much pure population of both primary and secondary, and each one of these represents the patients that we took. This is all acute febrile because what we are interested in is plasmablast responses. So the data I will show you here is about 170 clones that we made. About 100 and odd are from secondary dengue, about 67 odd are from primary dengue. We are making up the numbers. This is actually ongoing unpublished work. The only thing I want to point out on this slide is what you will see is interesting is that primary dengue is defined by IgM being greater than serum. But this is where I go back to a few slides before, where I had said that the serum gives you what has already happened and what is ongoing. It does not tell you what is happening at the cellular level. And you will appreciate that right here. While these individuals, their sera had IgM more or was only IgM positive with very little IgG, most of the clones that we made from the single cell sorts that we did were actually of the IgG phenotype, which means class switching in these individuals have already happened. And therefore, the cells have already started to make IgG in a few days to about a week. If you were to retest that person, you would probably see a stronger IgG response in their sera. So these are the number of clones we made. Most of them were IgG responses. So for the most part, for the rest of the talk, we will probably look at the IgG data here. So the summary of this part is we made about 100 odd monoclonals, 112 are from secondary, 67 from primary. And interestingly, the patient sera from primary dengue, as I already said, was IgM dominant, but the cellular plasmablast response has already switched to IgG responses. And both primary and secondary plasmablasts obviously have more similar IgG clones. So the first question as immunologists we are interested in is what is the immunogenetic makeup of these monoclonals, and which are derived from primary and secondary responses. Now, bear with me here for a few minutes. This may become a little too much immunology for some of you, but I'll walk you through it real quickly. The reason we are interested in immunogenetic makeup is that primary responses, one would expect, are primarily naive B cell responses. Secondary responses, as my cartoon slide showed you, we would expect more of a memory B cell response. There will be a naive B cell response that will come because in dengue, for the most part, you get heterologous infections, but that will be slower and smaller. And then later on, it will pick up. Right? So you kind of expect some level of differences between primary and secondary, as one is coming preferentially from naive, one is coming preferentially from memory cells. So the first question we wanted to know, what is it looking immunogenetically? But before I show you the data, one quick crash course on immunology in terms of antibodies. As most of you would know that antibodies are made of heavy chain and light chains, and the heavy chain has VDJ genes on it and the light chain has VJ. The diversity basically comes because the VDJ has about maybe 40 genes and diversity has 27 genes and joining segments have 6 genes. And depending on whether you're a light chain kappa or lambda, you could have 40 and 5 VJ or you could have 30 and 4 VJ. So permutation combination of all these VDJs and VJs results in a lot of diversity in the antibodies itself. And then once these VDJ combinations are happening, you either lose, replace, or change amino acids, and that results in further junctional diversity. So based on the combinatorial diversity of VDJ and VJ and the junctional diversity, you get a lot of immunoglobulin diversity, because that's how the immunology or the immune response functions. The other way you increase the plethora of antibodies that you make in your body is what we call somatic hypermutations. Now, this essentially is for the affinity maturation of the antibody. So once the B cell sees an antigen, germinal centers are formed, it goes inside, it gets T cell help, it goes in and out, and it cycles. And each time it does it, there are somatic hypermutations, as we call it. The reason we call it hypermutations is because the mutation rate is much higher. It's apparently 1,000,000 times higher than spontaneous rate of mutation. And what it does is essentially, this is what we show in textbooks, that the antigen kind of binds to the antibody, but not really well. But as the hypermutations are happening, the affinity matures more and more. And finally, you get a much tighter binding, right? So based on your diversity and your somatic hypermutations, you can expect functional differences in the antibody response. So with that quick crash course, this is what we saw. Now, I won't spend too much time on this slide because of the audience. The only take-home message I want you to see is that, again, these are secondary infections, these are primary infections. And again, this is divided by heavy chain and the 2 light chains, heavy chain, 2 light chains. But I just want you to appreciate the color coding right here and the color coding right here. Apart from a little bit of yellow here, and the yellow is absent here, if you look at the color coding of these 3 pies, which basically represents the different BH genes that the antibodies are made up of, there was not a lot of difference that we saw. So there were remarkably similar gene usage that we saw in primary and secondary dengue, even though one comes from a primary response and one comes from a secondary response. That means naive versus memory. Where we did see a difference was in the somatic hypermutations. So if you look at this, y-axis is percentage hypermutations. And what we did, we took all the clones, and the heavy chains are, each one of this is an individual monoclonal, and each one of this is an individual monoclonal. Similarly, the light chain here. And you can see there's a good statistical difference in the amount of somatic hypermutations these monoclonals have, if the monoclonal was from a secondary individual versus a primary individual. And this would be something that one would expect, because a secondary antibody is already coming from a memory that would have probably gone through a germinal center, would have probably affinity matured, and therefore has a lot of mutations, as compared to primary, which would be a naive B cell. So here we go to summarize this part. While the antibody gene usage in primary and secondary is remarkably similar, as I just showed you, primary dengue actually has lower somatic hypermutations as compared to secondary dengue responses. So the next question was, what does this mean in terms of antibody functionality? Because all these diversity and affinity maturation essentially takes charge of antibody functionality. Maybe it binds better to the antigen, maybe it neutralizes the virus better. So now that's what we were interested in terms of functionality. And we looked at the functionality in 3 different ways. One was regular ELISAs to see how they bind. Second was to look at its neutralizing properties. And the third was ADE. As I've already mentioned, in case of dengue, ADE plays a large role. So we looked at that too. So ELISAs, as I said, I've made cartoons for everything. It's a direct ELISA. What we do is we coat whole fixed dengue antigen onto our ELISA plates and just come back and enumerate the monoclonal by diluting out the concentrations. A simple, basic, direct ELISA is what we do. But we keep the coating as 1, 2, 3, 4, that we can not only look at binding of an individual monoclonal, but we can also look at the cross-reactivity of the monoclonal, because we know the infection that the patient had. So if it was a dengue 2 infected individual or a dengue 1 infected individual, we can walk back and we can ask, what does the antibody response look like in terms of cross-reactivity to the other 3 serotypes that are remaining? And here's what the data looks like. Again, blues are primary, reds are secondary. Each one of these graphs is dengue 1, 2, 3, and 4 that was quoted on the plate. What I'm showing here is OD binding binding to the antigen, and these are our dilutions. And as you can appreciate, in each case, the red lines are higher than the blue lines. And if you were to look at the minimum binding concentration of these monoclonals, if you translate these graphs into concentrations, statistically difference is seen between how well a secondary monoclonal binds versus how well a primary monoclonal binds. across all 4 serotypes. So both primary and secondary monoclonals are cross-reactive because, as you can see, that the blues don't disappear in any one of these graphs. So they have a capacity to cross-react, but the overall minimum binding concentration of primary monoclonals is lower than secondary monoclonals. What about the avidity of these antibodies? So avidity is basically the strength with which they bind. For some select monoclonals, we are actually doing things like BLI so that we get the exact KD value. But because hundreds of monoclonals have to be tested and BLI would be too much to do it, we did it the old school method of doing a urea wash. And the idea of a urea wash is that you do a simple ELISA, but if the antigen and antibody are not binding very tightly, as soon as you give something like urea, it will dissociate. And if it dissociates, the OD values go down. So that's a old school method of looking at avidity across two populations. And we did it for all our secondary and primary monoclonals. And this is what we find, that keeping in lines with what we saw, that their affinity SHM was higher of secondary monoclonals. They also bind better, that their minimum binding concentrations are higher. When you do this urea wash ELISA and calculate avidity index, the avidity of the secondary monoclonals is far higher than primary monoclonals. So all this indicates that while secondary infections are considered severe, there is an aspect of ADE and everything. At least the clones that we are making from the secondary individuals seem to look like really nice antibodies for the most part. But the true test of an antibody is obviously neutralization. So that's our next question. So everything is fine. They seem to bind better. They seem to have better avidity. But what does the neutralization look like? This neutralization assay, at least for these monoclonals, has been done by the focus reduction neutralization test. Some of you who are not aware of it essentially looks like this, which is shown on the right-hand side lower slide. We plate verocells, and then we come back with the virus and the antibody, the way it is shown here. So we pre-incubate the virus and the antibody, and then we add them to the verocells. And after incubation, you basically come back and you remove the supernatant and you fix the cells and you image the cells. And the idea is that each one of these spots is an infected cell. So the decrease in the amount of spots would be that the antibody is neutralizing. So I chose this picture to show you that in this case, this particular antibody neutralizes only dengue 2. So this is virus alone. So there is no antibody here. This antibody is unable to neutralize dengue 1, dengue 3, or even dengue 4, but it neutralizes dengue 2. And if you start seeing when does these spots go back to about 50% of virus alone, it is somewhere here. And then you calculate that this is its PRNT 50, right? So this is what we did for all those 160 odd monoclonals. And here's what we find. So when we plot this based on concentration into curves and start calculating inhibition of dengue virus to calculate PRNT 50s, here's what it looks like. Again, reds dominate the blues. So this is the concentration of monoclonals here. And if you take all these curves and calculate the PRNT 50 right here, and then look at what concentration it would be at, the graphs look like this. Again, in lines with what we saw for binding, what we saw for avidity, the neutralization capacity of secondary monoclonals seems to be far higher than primary monoclonals, which is all rather interesting. So everything that is happening suggests that when you have a secondary dengue response, you probably do have a protective response. But there could be a possibility that there is enhancement also happening, because that's the entire idea, that when you get a secondary infection, there would be antibody-dependent enhancement, which would result in severe dengue. So that was the last thing we tested. Oh, one last thing, that we also looked at the breadth, because we look at 1, 2, 3, 4, because we are interested in cross-reactivity. We did look at the breadth of the response by just dividing the monoclonals out into those that were not neutralizing versus they would neutralize only one serotype or two serotypes, 3 or all 4. In this case, the red part of the pie is all 4. And if you look at this red pie and this red pie, the secondary monoclonals, not only do they neutralize better, they neutralize across all the 4 serotypes. So their cross-reactivity is a lot better, not surprising. but it is, and it is significantly better. So this portion of the pie is a lot larger than this. Even 3 serotypes is slightly larger than this. So to summarize it, as compared to primary, secondary antibodies have better binding. So primary dengue have lower binding ability, lower avidity, and lower neutralization potency, and even lower breadth. Which brings me to the last part that I had just alluded to you, which we were really interested in, that are they even capable of enhancing dengue infection in vitro? So before I go there, again, with the idea that there may be few people in the audience who are not from dengue and are not familiar with ADE and not familiar how ADE is tested in vitro, I have a quick slide to show you how we do it. So the way ADE works, which I had just told you in the introduction, is that you have a target cell. Typically, it's a monocyte, basically a FC receptor bearing cell. And if a virus and an antibody is bound, and this is bound at a concentration wherein the antibody cannot neutralize the virus, then if it docks onto a FC receptor, it will actually go inside. The virus is allowed to facilitate its entry, and then it replicates, and there's increased viral load. To replicate it in vitro, what we need is a cell line that otherwise would not get infected with dengue virus, because that would not allow us to differentiate between infection, that for example, Vero cells get infected, BHK21s get infected. Those are not cells that you want to use for an ADE assay. You want to use a cell line for an ADE assay which does not have tropism to your virus. So it has to be a non-permissive cell line. So if I just take the cell line and I just put the virus, nothing will happen. Okay, so the virus cannot enter it because it's not permissive. There's no antibody, therefore there's no infection. But if I add an antibody, because this particular cell line, for example, in this case, we've used K562, has FC receptors, what happens here will get replicated. So the antibody will bind to the virus and it will go through the FC receptors, and then it will basically dissociate if it's a non-neutralizing antibody, and the K562 cells will get infected and you'll be able to see it on a flow cytometer. So here's what it would look like. This y-axis is arbitrary fold increase. When you have a lot of neutralizing antibodies, they will all bind to the virus and will not allow this docking process, or even if it goes inside the dissociation process, to happen. So all the neutralizing antibodies will bind and the K562s will remain uninfected. So you basically have no infection here. This is supposed to be infection on the y-axis. On the other side, when you completely dilute out your antibody, again, there's not enough antibody left to bind to the virus, take the virus to the FC receptors, or go inside. So again, because it's a non-permissive cell line, again, you'll get a flat line. So again, there'll be no infection. So on one side, there's a ton of neutralizing antibodies, and the neutralizing antibodies will keep the virus together. Thus, K562s will not get infected because they're not permissive. And this side, because there's too little antibody and you've gone back to this situation, since the cell line itself does not get infected, you get a flat line. But somewhere in the middle, where you have an antibody that does not neutralize very well, but can bind to the virus and do that uptake mechanism, you will then see an increase in the infection of K562, and you'll see this typical bell curve that is made. I hope that is understandable. So we did this for all our antibodies with the idea that hopefully, even though there is cross-reactivity of primary monoclonals, we won't see a lot of ADE perhaps in the primary monoclonals, and we'll see it a lot more in the secondary monoclonals. And here's our data for it. So I'll quickly walk you through it because we decided to actually divide them into two, because entire concept of ADE is the antibody is to be non-neutralizing, but it should bind. So all these monoclonals bind to dengue, but this side, on the left side of the slide, are the monoclonals that did not neutralize the virus, either 1, 2, 3, 4. And this side of the graph slide are the monoclonals that did neutralize the viruses. And then across the rows goes dengue 1, dengue 2, dengue 3, and dengue 4. The red ones are our secondary, blue ones are our primary in both the boxes, right? And the fun thing that you see in both the cases is that you can see these kind of bell curves in almost all the graphs. We were hoping that the blue ones remain flatlined, and it's only the red ones that show us that bell curve. But that's not what happened. Is a portion, not all of them in secondary infections either. You have a lot of them that are flatlined. A proportion of both primary and secondary monoclonals had the capability of doing ADE. That was one point of interest that we found. The second thing that we found is that both neutralizing and non-neutralizing antibodies were capable of doing ADE. And the third, even more interesting point that we found is because we knew these antibodies had come from which patient and what was the infecting serotype of that patient, we've also walked back, even though the data is not shown here, that we actually observed this for both homologous and heterologous serotypes. So all these three were very interesting. So what does all this mean? So if we look at secondary infection, they are IgG greater than IgM. They check off all the boxes of secondary infections, and you take all of them and you make individual monoclonals and start asking what individual B cell antibodies look like. This is the summary of all that I have shown you. They are very strong binders. Many of them have very high avidities. They are also potent neutralizers. They have high stomatic hypermutations. They are cross-reactive, both in terms of binding, in terms of neutralization, and they can also enhance infection. This part unsurprising because all of us have heard of that. The surprising part comes during primary infections. Now, if you take primary dengue, make human monoclonals and ask exactly the same questions, we see low binding, we see low avidity. They are poorer neutralizers. It's not like they don't neutralize, but they neutralize to a certain lower extent as compared to the secondary monoclonals. They have lower somatic hypermutations, which is not surprising since it's a naive repertoire that is expanding. The interesting thing is that they are cross-reactive. So for the most part, at least in literature, when you read about primary and secondary, one thinks that primary responses are more towards a serotype specific responses. We didn't see that. We actually saw cross-reactive responses. We actually even see that in sera. But more interestingly, they actually both homologous and heterologous serotype. So what does all this mean? So I think here is what our bottom line is, that we think that we are not like trying to say that something is wrong or something is right. Obviously, severe dengue exists. And what we see is that severe dengue is actually not just exists in secondary infections. We also see it in primary infections. The caveat being that we are looking at hospitalized patients. But what we think is that while secondary infections may be contributing to severity through processes of ADE, primary infections may be contributing to severity through diminished protective immunity because they are lower binders, lower avidity, poor neutralizers. There's a possibility that by the time the protective immunity takes over, there is a chance for the virus to do whatever else he does. And when we put all this together, simplifying everything, this is why we see this phenotype, that we see equal proportions of primary and secondary dengue, severe disease and primary and secondary dengue. So this is my last slide in terms of the data I wanted to show you. But the only point I want to make here is before we move on to the acknowledgements is that what I have shown you is B cell responses and antibodies. But as a scientist, I do want to acknowledge that this is a little bit of a simplified view when you want to look at only one thing at a time. As one of my introductory slides, I had shown you all the data that we are gathering: innate immune responses, T cell responses, serum antibodies, what type of virus is there. I want to acknowledge that getting severe dengue is more complicated than just what your antibody response looks like. So while we've not been able to see that, secondary infections have these kind of antibodies and thus this entire thing is happening, and we've not tied a neat bow around it, we've gotten to one part of this puzzle. But there could be other contributing factors to severe dengue, just the host, the T cells, what kind of cytokines the T cell make, what kind of virus was there, what strain of virus was there, some other HLA makeup. There could be a lot of things happening that could be resulting in an individual developing secondary dengue, including the kind of clinical care he got or what time the clinical care came. So there are a lot of factors to keep in mind. I just wanted to put that out there in the end rather than leaving it that, aha, we've solved the problem. So with that, I'll stop. And I think I am a few minutes beyond 45 minutes, but I really couldn't have shown you this data without the students who actually do the work. Charu is the one who did all the work on the plasma blast and the cellular responses, and she's the one who's done all the transcriptomics. She's now graduated. Pragati was the one who originally made the human monoclonals and started this work. Most of the work is actually hers that I showed. But since we are continuing this work and we're in fact making antibodies from now the same individuals when they recover, what do they look like? And Ram and Keshav and Sanjeev are a part of that work. So I've shown you work from all these individuals. I'm very thankful to my lab. I have a very vibrant, fun lab that I work with. And as this lab is a partnership between ICGEB and Emory Vaccine Center. This lab or this science obviously is done very closely with the director, Rafi Ahmad. Murli, I work very closely with him. He spends half his time here, and Jens and Carl are actively involved in this work. And obviously the funding agencies. With that, I'd like to say thank you. And I hope you understood what I had to communicate, and I'll be happy to take any questions.
Thank you, ma'am, for that detailed and very enlightening talk. It was an honor and a privilege to have you with us today. So now we'll begin the Q&A session. If you have any questions, you can unmute your mic and ask the question. Any questions?
Yeah, this is Darshan here. Can you hear me?
Yes, we can hear you, Doctor.
Yeah. Okay. Yeah. So just to identify myself, I'm Darshan Disil. I'm a PhD scientist at KDU, and I work with Alessandro Setti, Aravind Disil. I'm sure you're familiar with them. Yeah. So very nice talk. I was actually.
We have their mega pools.
Yes, good to know that. Yeah. So I was quite impressed by your data. I just wanted to point out something. When you did the last set of assays you were doing, when you were making your dengue virus, I'm not sure if you've seen this paper. So actually, I was part of that paper. It came from Aravinda's lab, and its first author was Raut et al. In PNA's paper, where we showed that sometimes when it's something I had suspected all along, and we didn't have the facilities here. So Aravinda's lab, we did it. Raut did most of the work. Where we showed that actually viruses, the dengue viruses, when we make it in the lab, the cell culture, it's quite different to what we see in the human. And that paper outlined it, and Scott Halstead had written about it also.
Yes.
What did
very
you
famous paper.
Thank you. So what did you do to get around that problem when you're making viruses?
So honestly, nothing as of now, because those are the caveats that, yes, I completely agree with you exist. The only thing we are being able to do is make sure that we don't use mosquito virus, because there will be no glycosylation in C636. So we are using Viros to grow virus. We try and make sure that we use the temperatures that are needed. We also have, we do sucrose gradients to make sure we have as homogeneous as possible live virus preps. We also try and make sure that our dengue virus 1, 2, 3, 4, when we infect them just like that in Viro or BHK, give us the same, more or less same amount of infection at an MOI of 1. But with that said, dengue virus can be very finicky, and I hate dengue 3 for that. It just dies. Considering keeping that paper in mind, I completely agree with you, considering that, dengue 2 can breathe versus dengue 3 can do something else. Our capacity ends there. But what I wanted to add to this is that we have a really lovely collaborator, Guru, who's a virologist. So we are working with him for at least a select few monoclonals as a part of another program, to see whether when we talk about cross-reactivity, we talk about neutralization, we talk about ADE, if a virologist does it, making viruses the way he would make them, and different clinical isolates, how much of it holds true. So does that kind of answer your question?
Yes, it does. Thank you.
Okay.
Yeah. So,
Yes, but I
if
wish
can,
someone would come and just make lovely viruses for us.
yes, I know it's something I have dreamed of, but it's just been beyond some of our capabilities here. I wondered if we took, if we had, say, human lymphocytes, if we could get, from human healthy cells, if we grew it there, whether we could get something, which is better. I don't know if that's even practical to do, but and how we could maintain the homogeneity and all
So
that.
I think you've touched that thing that having a homogeneous culture is very important when we start doing such assays, which is why we stuck to reference strains. But as a part of one of the other projects that we have ongoing, which I said Guru, the virologist, he's at THSTI, which is in Faridabad. He is making clinical isolates, and the way his clinical isolates are made is from whole blood. But obviously, they will eventually go into viros and get expanded. And so, getting the exact same virus from the human, I don't even know where to even start with that.
Okay, thank you. If I can have one more question, I'm not going to get into the T cell part since I'm sure you're familiar with Danilas and all that. Now, in Sri Lanka, we have seen this consistently where when we have a high wave of infections, we have a different strain coming in and we are clearly able to see that. Do you actually believe that one strain can come in and dominate and cause something like this?
Can I give
Thank
you a
you.
personal call and then we'll talk about that? Because
Sure,
you know,
sure.
there is this thing of, yes, every 3 to 4 years you will see a wave that is heightened,
Yes.
and we see that in India also, but India is much larger, right? So what
Yes.
happens is that the transmission seasons are very different. So now we are going past what would be immunologically the reason versus what is the virus doing versus maybe the immunity goes down. So there is, it's not as simple as what A goes to B. There are lots of little complications in the middle. So short answer, yes, we also see dengue coming in waves, but what we saw from 2012 to about 2015, 2016, we continuously in Delhi kept seeing dengue too. Whereas in the south, where we were collecting samples as Senj, whether that is because most of our studies were in hospitalized settings, because it's just easier to do them when you especially are looking at such immunology, or whether this was a true reflection of what was happening in the population is something that I cannot answer. But does the wave come up with heterologous is also further complicated, because you have to have very close surveillance. Whenever you do such studies, you need a lot of surveillance of the type of serotype, and somehow the surveillance is done in pockets. My data is my data, and then I have a virologist who then looks at only these 100 individuals, for example. So while I agree with you that we see what you see in Sri Lanka, there are differences just geographically. How long is the transmission season? Which serotype is circulating? So, for example, now we are seeing dengue 3 in Delhi for the past maybe 2 or 3 years, and that has not changed. It'll be interesting that in the 4th year now, suddenly, if another serotype comes, do we see a wave again? But severe disease we see every year. But that could just be hospitalizations. We are getting our samples from referral hospitals. The way India is set up, if I have an infection, I first go to a local clinic, then that does not help, then I go to a slightly bigger guy. If that doesn't help, then we get to an All India Institute of Medical Sciences. By then, I've lost that critical window period where I could have got clinical help and things could have resolved. So we don't know our severe disease cases, which is what I pointed out on the last slide, that while all this looks well and good in immunology, in human infections, because you don't know T0 and it's not controlled, there are a lot of these kind of factors that also we have to keep in mind, and we cannot just draw parallels straight away.
Okay, thank you. We'll certainly keep in touch. Yeah. Probably I should fly out there one day. We can have a longer chat. Okay.
Yes, that would be great.
Thank you very much.
Okay, any other questions?
I see a question in the chat box that asks,
Yes.
what about
yes.
infection related to third or fourth infections? We don't know. So our studies are not currently capable of following the same individual. To be able to say third or fourth infection, all the individuals, for example, we have as primary infections in the first set of graphs that I showed you, I should have the capability of following these guys out, and I wish we did. Following these guys out, even if they never develop symptoms, that's the most important fact. You know, even if they don't develop symptoms, if we could keep getting a drop of blood to see even if they got asymptomatic infections, and then we would have some idea about third versus fourth infection. When I speak to my clinicians, because I want to know about these quaternary infections, for example, they collect history. And based on history, they say that they rarely come across people who say that I've had dengue 2 times, 3 times before and confirmed dengue. See, that's another problem. That did you have confirmed dengue? They may have had symptoms, we don't know what they were. So third, fourth infection, I literally have no idea. Unless and until we invest in some really good population studies where we follow cohorts of individuals throughout the year, including transmission season, over a period of sustained time, will we ever be able to answer that? That's a great question, but there's no answer, at least not in my lab.
Okay, thank you. Any other questions? And one last question, ma'am. Why is it so difficult to develop a vaccine for dengue? I mean, it has been such a problem in the world, but we still don't have a vaccine. Why do you think it's such
wow, that would take another 45 minutes, right?
Okay.
So one of the complications is like this primary, secondary infections, right? So now when you have 4 serotypes, there is a compounding and a confounding factor of to be able to develop immunity to all those 4 serotypes to the same level to make sure that antibody enhancement does not happen, all those kind of things. So while there are vaccines, and there are vaccines that are going through clinical trials, at least the one Sanofi vaccine, for example, a large reason for its failure was that they did see ADE. Individuals that got vaccinated some of them developed severe disease. And so it has been approved only in people who already are seropositive. So that is one major reason itself that you have to be very careful when it comes to deploying a vaccine wherein you have multiple serotypes at play. So if I was to simplify everything and not take another 45 minutes, I would say something as simple as that. One needs to be extremely careful. One of the things is being done in Naveen's lab. Why don't we identify what results in neutralization? Why don't we take only those portions of the virus and leave all the other things out? And that way we can perhaps clean up the system, and give people only what they want and develop neutralizing antibodies. But that would mean that we are restricting that vaccines are only neutralizing antibodies. If one thing even COVID has taught us, that vaccines, all the empirical vaccines also work best when you have both T cells, B cells, innate responses, all of them working together. Immunology doesn't work in a box. It's not T cells sitting here, B cells sitting here. Everything has to come together in an orchestrated manner of things going well. So till we basically understand what human immunity looks like and what do we need in all these arms of immunology, I think we'll eventually have a vaccine.
Yeah. Okay, thank you. So then I think we'll wrap up the session because it's past 12 o'clock. So once again, ma'am, thank you very much for your valuable talk. And it was an amazing, great talk. And thank you very much, everyone, for participating. I hope all of you enjoyed the talk. So goodbye.
Thank you so much for having me. Really appreciate it.
You're welcome.
Bye.
Bye.